there was too much of the sediment other than the coral rubble, but
it's quite hard to collect sediment with
Yeah.
a claw like
with
that.
a claw.
Not
Hoping
the
there's
right
some
tool for the
joy
task.
lock.
in between some of those rubble pieces, but
And
not
I'll
too
push out
likely.
ahead because I see we're behind. All right, watch these. I just need to do a small push for a second. We kind of fell behind a little bit with Sirius.
Roger that. So we're going to move along this mound for a bit here and catch up with the ship a bit, since we were sitting still and the ship was still being pushed by the current. But we'll get some really good views as we fly over. See some more, probably an Elipsamia there on the left. More sponges coming into view.
Copy that.
Well, I think it's pretty clear that we've confirmed that these are certainly
So
habitat.
you look like you're following the ridge. And
This
you're
is interesting
due
on
north,
the right.
So
We'll
Yeah.
have to take a look
co-pilot,
at
I
this.
think our next move is going to be over that direction.
Yeah, to my left or port is
That
down
would kind of
slope.
agree with Dimitri as well.
These look
We're
like some kind
turning
of
back
burrows.
north
Oh,
to the
great.
east.
Right.
All right, so
Somebody else might be here
yeah, bring your
on the
head
very
around,
top of
kind
this
point
mound.
towards D2 if you would.
So some of the fish species that are commercially fished in this area are tile fish, and I know that they make burrows, so I don't know if they'd be this deep, but I guess it's a possibility.
And I would go with that heading, so that's yeah, 0. Looks like it's still off a little bit to your port, but all right, co-pilot, I'm going to take you 010.
And
Does that
then
sound
Pilot,
alright
can we
to you?
just take a quick zoom on those burrow-like features or mounds? We can't even tell.
do you say mounds, watch it?
Yeah, right
Yeah.
in the center ahead.
Copy.
Well, actually,
You can see that fresh sediment on top
quite tell
in these
that's
mounded areas.
uphill.
That's what
Yeah, it's
underlies
uphill.
this
So let's bring
slightly
you over
brownish
starboard some, actually,
overlying sediment.
because we want to stay on top of
The
this
brown
ridge line.
tint of the
So we'll do that heading.
the top
040.
sediment
All right,
is
video,
from
go
that
ahead,
iron
zoom in
manganese
on mound.
coating on each grain. We'll be able to see this if we get a sediment sample, the difference between
Bridge,
surface
I'll be there.
sediments and these pieces.
Great, thank
You
you.
also see it on the rubble of the coral,
ahead,
the dead
RV now.
coral stalks.
Ship move,
And so the
please.
white
30 meters,
dead coral has been buried and
040
recently
at
exposed.
Has
0
not been
decimal 2.
exposed, yeah.
0 meters, bearing 040.
Yeah, you
There's
can
a
see
tiny
some-
crinoid.
And a
Good copy, thank you,
pteropod
Rich.
on the sand. You see shell remains of pteropods, the planktonic snails that
Oh,
our
yeah.
colleague Stephanie
Sorry,
Bush
video.
of the Smithsonian, that's her specialty. I hope she's watching. But these pteropods, they look like little winged
ROV now, Bridge,
organisms,
moving
which
initiative.
is where they get their name.
Thank you,
Wing
Bridge.
foot is what they are called, which is actually the soft part. Has a foot-like, is a foot-like shape, and helps them motor a little bit. They still are at the mercy of the currents, but they can maneuver somewhat when they're alive.
All right, watch leads. Are you good with that?
And Stephanie's letting us know that it's likely Cavalinia. That's great. We will try very hard to get a sediment sample, maybe as an associate with some organism that we collect.
Is this a sample? Watch it.
I don't think that we'll be able to
Yeah, I
get
think we
that.
can keep going on.
Yeah.
All right, copy that.
Thank you, though.
All right, video. Go ahead and come wide when you're ready.
But you can see that a lot of that material is quite coarse and riddled with shell material, which is what we would expect to see under the Gulf Stream, the finer materials that might be settling down from
Great,
above.
thank you. And then if you want to
Just
zoom
can't
past skids,
even
you can feel free to do
deposit
so.
on the seafloor. There's too much energy here, and they're too small. So we tend to see larger grains
We're going
in a high
10,
flow
or
environment like this. And those grains in this area are
040,
mostly
copy.
from
Yes,
the
well,
shell
come on, go ahead.
material of organisms, such as pteropods and
So
sponge spicules. There are many of those in the
we
area.
are heading
From the
currently at
sponges, they're
040. I'll
very
point that
sharp,
direction.
silica-rich
About right there
sponge
is the general
spicules
motion of
are
ship.
very plentiful in these types of
And our
sediments.
sonar is kind of pointing in that direction as the higher slope. So yeah, slightly to the right as I was looking is a little higher.
We also see a lot of planktonic foraminifera shells in the sediments, the larger ones, and these settled from the surface waters after the organism dies.
Yep. Copy that.
They're large enough
So what's
to
it look like,
withstand
90
these
degrees
currents,
to your
I guess. They're heavy enough also.
To right?
Yeah, if you just turn your head in.
That's where it came from. That'd be downslope, I think. So
If you've
that
joined us on
is
any previous dives,
south,
the seafloor sediments
Okay,
were
so that's
very,
south.
very small
Which is
particles
opposite of
because
Blake
of
Plateau.
the lower
That's where we
velocity
came.
currents,
we just came from
clay-sized particles, and we
So watch it.
characterized
That's south we're looking now, and that's where he came from.
that type of
So we
sediment
could-
as being mud.
We'll,
and
do we keep moving towards
really
waypoint
don't have
3-ish
mud
though?
here.
Yeah, let's continue on the bathymetry that it showed a dip in this region. So I think we're right on
Right,
track.
so
but
yeah.
East-west,
still-
we're on the local high.
We still have a slope this way, so
Yep.
yeah, that's great.
In another 50 meters or so, I think we'll be heading uphill again, if that.
Copy.
Go ahead. So we've traveled about 70, 80 meters based on our on-bottom point, but the ship we've put in 120 meters of movement with the ship, but D2 has only seemed to have covered about 80 meters on bottom. Roger that.
So just for perspective,
Also, it's about halfway.
we've
Does
moved
either
about
one of you
80
want
meters
bottom
since we've
terminal?
reached the bottom, and we've seen all sorts of interesting coral habitat.
Yeah.
The
So-
ship is
sorry,
moving a
what'd you
little
ask?
faster than
I see
we
either
are
one of you
in
wanted
the
bottom
Gulf
time,
Stream.
but Andy answered first, so I can't give you any.
And we've moved off the top of the mound into a slight depression here, and you see an immediate change in the
Watch
seafloor
the
character.
snap.
I can set real quick. We can switch.
So, accounting for some of the backwards over ground motion we made right after we came on bottom, I would say we've covered about 100 meters at this point.
And after a little bit of calculation, it looks like we have covered about 100 meters now.
All right, Copilot, you want to
So-
hit joy lock real quick? Sorry.
And our dive track has close to
Pilot, why don't
700
you, actually
meters
keep driving for a little
altogether.
bit?
Okay.
I need to see if I can
Planned.
find someone to help me disinfect my station before I hand it off to you.
And that would basically cover at least
Oh, I could go
two
to
of these mounds.
Copilot.
And the last one being quite a bit larger than the one that we're on now.
Oh.
Well,
yeah,
we
sorry.
actually started
Yeah,
on the
whoever-
top of the
Yeah, you
first
just
mound.
drive
Yeah.
for 5
Okay.
minutes
So
and then find someone from the dry
We've
lab.
finished that
Sorry,
first mound, and we have
misunderstood. Yes.
2 mounds left, each of which is increasing in size. It'd be nice to get to that top, steepest and highest mound if we can.
Right.
So we might try to get some headway here. And you'll note that as soon as we got off the top of the mound, we stopped seeing the abundance of corals and sponges, and certainly less rubble. It's probably just beneath what we're looking at. This is where some of the sediments have been able to deposit, and coral hasn't populated in these areas.
Yeah, watch it. This seems to be a little dip in the terrain, but our sonar is picking up slope just ahead of us, the way we're moving, so we should get upslope in just a moment again.
Roger that, pilot. So pilot
yeah,
has
you can see
confirmed
it.
that we just are going into a dip, but that there's another upslope in front of us, and you can just start to see that now.
fairly steep.
Mm-hmm. And even in this dip, you see a little bit of coral rubble. And maybe a couple of stylaster corals there, the little white guys.
Maybe a quick zoom, please, pilot.
Yeah. All right, video.
Little tiny white guys.
They are.
They're really hard to see.
Yeah, these are.
Yeah,
The
stylasters.
Now, is that one of the stony corals?
But that's a
not?
hydroid.
Okay.
Hydrozoan
Oh, it's
coral.
not even. Okay. They look so white. They look solid.
Yeah.
Good.
Yeah, that's great, pilot. Thanks.
All right, video, whatever you need to do, you can go ahead. And then when you're ready, you can come back out. Great. Something ahead. Yeah, I'm heading there. Oh, video soon from here.
We've got a skate here coming up.
Casey flies away. Copilot, please kill lasers.
Huh, look at that. It's quite a beautiful tail.
Beautiful tail, yeah.
You had enough of us.
No, we're just getting a little too close for his comfort.
All right, video, you can try to push again.
Mm-hmm.
These skates are cartilaginous, similar to sharks.
Yep.
We've not yet seen a shark. We often get that question.
Yes.
Have you seen any sharks?
But this is probably the
All right,
third
video,
ray
can
that we've
let 'em
seen,
go.
maybe? I think we've seen about 3. None of them have been huge, but this looks like a different one compared to what we've seen before. Of course, we were much deeper when we saw the other
All right, Copilot, I'll center with you.
rays.
Skate is following by coincidence. Pass him up there. I think I could
Wow,
Pilot,
look at
whenever
this.
you're ready to
Look at this
Yeah,
mound
swap,
coming
I
up.
think
That's
I've got
amazing.
most of it over here.
Yeah, stand by. Let me just
If you want to hand off in the water, you can.
gently and set real quick. Make
Okay.
it a little easier. Stand by 1 sec. Copilot. All right, go ahead. Great.
Watch leads, pilot.
Go ahead, pilot.
As we do our swap here, I realize we never got a chance to introduce ourselves, so now's a good time.
Oh, that would be a great time. Thank you.
Well, hello on shore. This is Bobby Moore. I just swapped into pilot. Your previous pilot to my left, now sitting navigator.
Sean Kennison.
And to my right.
I'm your ROV co-pilot, Andy O'Brien.
On video, I'm Emily Narrow.
And on clipping, I'm Annie White.
And we are all engineers and videographers from the Global Foundation for Ocean Exploration. We're tasked with managing the ROVs and video systems, highlight videos, satellite systems, and most other technical aspects of getting down to the bottom of the ocean here.
So in other words, they hardly sleep while we're out here.
And we apologize for the lack of introductions.
We'll
That's
do
what
it.
happens when we land and we immediately start to see something.
Yep.
We forget our manners. So thank you for reminding us, and we appreciate all you do. You're an incredible team, both the ROV team and the video team.
Okay,
Amazing
stand
group
by
to work
video.
with.
Gonna come around a little. All right, can start to go in slow. It's quite all right. Sure.
Pilot, can we get a quick laser when you pull away from?
Roger that. Hit my toe. Come around.
Don't want to blind the poor fellow. Oh.
Yeah,
Beautiful quick shot anyway.
Yeah, let's come wide. He doesn't quite want to stay in our lights.
We're coming up to much
This is your
more
final
coral.
one if you want
Looks like
video.
we have a bunch of bamboo corals on the left.
No, he's not going to comply. All right, let's come wide and move on.
And Pilot, this is watch one.
Go ahead.
Could we look at these soft corals, these bamboo corals on
You
the
got it.
left-hand side of the screen? Thanks. So they're all over the place, I guess. Wow.
Is there one in particular?
Whatever is your choice, whatever's easiest.
All right, we'll go with the one center with a little darker background. Go ahead, video.
I hope Scott is still here. Scott France, he said he had to dash off to a defense, a thesis defense, but maybe he's still
stand
here to
by.
tell us about these bamboo corals before he leaves. You see some a shrimp on that
Still dialing
coral
in.
skeleton there on the bottom.
Go for the one on the right here.
It looks like we have an anemone growing on the bamboo coral in the center there.
Oh, copy.
And you see one polyp is closed up on the left there, and the rest of them are open.
Oh, lost my tone.
We have some cup corals in the background, those
Oh,
pink
sorry.
corals, and a nice big
Going back
hydroid
to
on
it.
the right that we just looked at.
All right, go ahead, video. Let's go back in on the right one.
And Scott France says that these are likely a type of Acanella. Although no doubt some would want to call them Isidella.
We're going to get clouded out here pretty quickly. Yep.
And what we're looking at is the branching pattern,
Let's come wide.
and it looks like it's branching just post-node and
think
not
that's
at the
enough for
node,
a
which
snap.
is the way that you would tell those two genera apart.
Is the yellow behind it, is that a coral? Doesn't look like similar to the sponges we've been looking at.
I would imagine that that is a sponge. You're talking about on the right side?
No, behind what we were just looking at video. It'll start to come in once I get this dust off my vehicle.
And I guess from that branching
Start to
pattern,
go in right here.
even Scott France is stumped about whether this is Acanella or Isidella. And it's going to remain a mystery because Scott has to run out to a thesis defense.
Okay, yeah, that's not looking like a coral behind there. All right, I think we can come wide.
But he is leaving us with his best guess, which is the B clade of Keratoisis for that bamboo coral that we're just looking at. Wow, and that's
auto headings in.
a very large
That explains a
skeleton
lot
of
it.
Analopsamia in the center top there. Pilot, this is watch one.
Go ahead.
could we get a look at that pink coral that's just center now?
You got it.
Thank you.
All right, video. Hopefully I'll do better now without my auto head pushing me everywhere. Start to go in.
There's a cup coral and more bamboo coral here. Ooh. And that's a coral we haven't seen yet today.
Trying to creep around just a little.
Pretty.
It's got a golden color skeleton, which is characteristic of the chrysogorgids, but I don't remember seeing one that looks like this. Looks like there's an anemone on the right side. And a beautiful cup coral in the back, and hydroids.
It's all yours, video. Okay.
Beautiful video here. And Chris Kelly says
Trying
this
to get
might
you
be
over
a
to the right
primnoid.
side to see the skeleton.
And Rachel Bassett, she and her team agree it's a primnoid. Some brittle star arms you can see there, the pink guys.
Is that good, watch leads, for this?
Pilot, can we pan just a little to the right, and then focus on the distance? There's some spines. I don't know if they're legs of a crab or they're down on the seabed.
On the seabed.
To the right, or if it's pencil urchin or nothing. We saw a pencil urchin earlier. I was hopeful. Okay, we're good.
All right, let's come wide.
Oh yeah, it looks like there might be a squat lobster to the left lower side, the reddish organism.
Do you want a close-up of those?
Sure, if
Yeah,
you
we
can,
haven't seen
that'd
one
be
yet
great.
today, so that would
And
be
we'll
great.
go to the pink in the upper right.
Yeah,
So let's
and
first go to squat lobster.
Okay, and then maybe the sponge in front of it as well. Have we seen that?
Yeah, I would imagine that's a Munidopsis of some sort. And then, wow, that sponge is definitely different than anything we've seen today as well. So Munidopsis are Galatheid anamurin crabs, so they're decapods, but they're more closely related to hermit crabs. They actually have 5 pairs of legs, but the last pair is modified
Sorry,
and
video,
tucked
I'm pretty
underneath.
bouncy. Let's come out a little.
And in hermit crabs, those are used to hold on to the shell. And Steve Oshkovitz has noted about that previous coral that we looked at, the primnoid, that it's reminiscent of several species in the Narella genus, which have a golden axis.
Oh, this is beautiful.
Wow, it looks like there's a anemone and a hydroid and a stylaster and crinoids and sponges, stock sponges, and the agglutinated forams even in there, maybe?
Sorry,
Wow.
video, I'm not doing very
Oh
well
wow, and
for
then
you.
there's also an octocoral, really small octocoral on the left
The
there.
long stemmed.
Yeah, the
one.
long stemmed. I don't know if that's large enough to even be able to tell what it is.
All right, let's come wide.
That's a stylaster. The small
Stylaster.
white coral that you see on the bottom there was a stylaster like we've been seeing previously. And Chris Kelly was asking, what is that sponge growing on? It's growing on the dead Analopsamia coral
Let's go in
matrix.
here.
I believe. And then we have, wow, these are a couple of Anthomastus, I believe. There's the red octocoral that you see in the center. And then there's also another octocoral, and then a glass sponge on the right.
With a brittle star at
brittle
its
star.
base.
Yeah, and,
Oh,
and
yeah,
then
look
some
at the
zoanthids.
Yellow this time.
Yellow.
We saw white zoanthids farther out to sea.
Yeah, so these zoanthids are growing on the glass sponge, and several people were commenting that that's actually a pretty common association, are these zoanthids growing on the glass sponges. And there's a crinoid and then a whole bunch of cup corals in the background, and more bamboo corals.
And even though these zoanthids look solitary, they're actually colonial. They're connected through what?
They have basically just a connecting stomach piece of tissue or organ. And it looks like a little bamboo coral there. Yeah, so Simon Brooke was pointing out that those sponges that we were just looking at that are on stalks, they're pretty common in this area as well as the Gulf of Mexico, and for lack of a better name, we've been calling them lollipop sponges for years. But maybe somebody
That's
has,
a good name.
yeah, maybe somebody such as Chris Kelly or somebody else might have a more proper taxonomic name for them. Wow, what a abundant little patch of dead coral that was.
And
Abundant
if you're
life.
just tuning in, we're on top of a mound that is representative, we think, of hundreds of similar mounds that cross this area. We're at water depths roughly in the 700-meter range. And these mounds stick up from the seafloor that is very flat everywhere else, and right under the Gulf Stream.
This one looks a little different
so
than what we've seen
top of
video.
one of those mounds, and
yeah, that'd be great to take a look at, Pilot. Thanks.
Dark branching material is dead coral that has been encrusted with iron oxides.
Another move in, or
and is being used by
great.
many,
I'll just do
many
another
different
30 at
organisms
040 degrees.
The hard substrate that they need to hold on to stay in place under this very strong current.
Go ahead, video.
And likely this entire mound is built upward from piling up over the thousands to maybe millions of years. We don't know the ages of these.
I am. You're very quiet.
There's several brittle stars.
But I can
Look
hear
at how tiny they are.
Oh, wow.
Do you want to try fog lights video?
How's that?
Oh, that's way better.
Great.
That's
Okay.
amazing how many there are.
0 meters at
Let's give it a go.
040 degrees.
Ooh,
Speed?
all right. Is that better or worse?
Okay, 30 meters, bearing 040.
There's
speed
also a
decimal
little
shift.
anemone down there, the purplish-looking organism.
Beautiful color. They are just decorating this, aren't they? And the thinking
Try
is
to
that they don't
tilt down
feed on the
gently.
coral,
ROV
that
now,
they are
bridge
just
move
using
initiated.
the coral to get high up into the
Oh,
current.
look at
Is
that.
that correct? So that,
Yeah.
oh, look how,
Look at
my
that.
goodness.
Is that a little eel?
It looks like an eel.
Wow.
Look at the tail at the base of the coral.
I've never seen anything like that.
Is that a worm?
It might be
That's
a worm.
mouth. Yeah, at first it looked like an eye and a beak.
Yeah,
but
it was
it's
turned
really, I
differently.
think, just a mouth. But it's moving.
The coral might be a plexurid that we're looking at, but I'm not sure what that is. Oh, there's a little shrimp on the end to the left. It's a hydroid that's below the coral on the left.
That is bizarre.
And if anybody
looking.
knows what this worm is,
It
would
does
be
have a tail. I mean, the end of it is at the base of the stalk. That is a very strange thing that will appear in some sci-fi
yeah, or my nightmares
movie.
tonight.
Yeah, I know. I don't care for that particularly.
So Nolan Barrett asked if this is an ophiuroid nursery or are these mature individuals? Oh, and Chris Kelly said that this
Pilot's
is a
clear
mollusk
when
that
video's
feeds
clear.
on corals.
Well, it certainly has a mouth
Roger that.
adapted to feed on a coral. Its mouth is
Yeah.
about the size of a polyp.
Got it.
That's just creepy.
Tilting up gently.
And Nolan Barrett said that it could be an Aplicacophoran. Aplicacophoran.
Aplicophoran.
Okay, yes.
All right, let's come on.
And then
Apla.
Megan
get
McCullough
fog
said
lights
that
off
it's
and,
not that.
Move on.
But then Mike Vecchio says it is. So
I'm
if you're
glad we're-
listening, you understand that we certainly don't know everything about the organisms that inhabit the deep. And there's another
Looks-
golden
yeah.
crab.
Oh yeah, another golden crab. More
There's-
dead standing coral.
Look off to the side.
Nice big sponge to the lower left.
So we're definitely on a little ridge here. We'll
And
keep
we've
following
been
you.
also going back and forth in the chat
Maybe
room
we could
about
take a
what some of
quick
these
zoom on the
sponges
golden
are.
crab.
You got it.
Thanks.
We were debating whether it was Africalistes, that was the sponge we were looking at that had the yellow zoanthids. And Sandra Brook thought that that's what it was, and then Chris Kelly thought that it was not that because of they don't branch like that. So there's still some back and forth about what that is.
There's
Okay, let's go
several
ahead
of
on the.
Oh yeah, we have
what are these called?
Anthomastus, or the
Anthomastus.
red corals on
That
the
were-
left. And here we have another golden crab. It's our second one we've seen today. The other one was kind of tucked under a dead coral mound, but this one is walking across the dead coral surface. More flat area.
I'm bringing them back. Stand by.
We have a little round brownish sponge and some round greenish sponge and some clear ones.
Pilot, can we put the laser on?
Lasers, please.
Just- yeah. Thanks. We saw one earlier that toe-to-toe is about, what? Oh no, that was the eel. So here the carapace is just under 10 centimeters.
Did somebody just join us on the phone?
Gonna try stabilizing you. Give you a good shot. Standby video. No, you're good.
Hey y'all, this is Trey. We have a group here at the Beams Biz Lab.
Hi, Trey. Trey Gillespie.
Yes, ma'am.
Do you have a question for us? We'd love to hear from you.
Awesome. We'll send any questions in this way. We're watching the video live right now.
Okay,
Coming
great.
left and up a little.
Trey Gillespie is a senior at the College of Charleston. He's a geology major and leading a team of other geologists. Who's with you, Trey?
Few seconds and hopefully this dust will clear.
And this is Chaseon, the golden crab that we're looking at.
It's Robin Humphrey's class, Geo103.
No kidding.
We're here checking it out, learning a little bit.
Hello, Robin. And everyone else, and welcome to the Visualizations Lab of the College of Charleston.
ROV nav bridge move complete.
We're looking at a golden crab.
Let me know when you're clear, video.
Hi,
All
Robin.
right.
Looking at your live feed
Yep.
here.
Yep. Time to move on.
Excellent.
Probably have another minute or two to swim.
Moving
Yep.
on.
They're very fascinated.
So that golden crab is one of the commercially fished species in this area. So it's really
Forgot to tell you
interesting
that
to see them
1449
and
is
see
where
how
I
they might
marked
be
the
using
first
coral
100 meters
habitat here. And again, that's
of
one of
transit.
the uses of this area. This is a coral habitat
No,
area of
of
particular
ROV
concern.
Making the
The
100-meter mark.
crab fishery it fishes all around the outsides of this area. They would like to use more of this area within
This is
the
new
coral
video,
habitat
so
area
we'll
of
take a
particular
snap.
concern. Oh, and there's a black coral too, Bathy Pathies. So anyway, okay, sorry. Yeah, so any information we can find about how much coral area is actually here and how the crabs may or may not use the coral habitat is information that can inform management.
Sounds like we got an idea on that. So let's carry on. Thank you. Tilting up.
And we saw the Bathypathes we were just looking at. We saw many of them farther out on the Blake Plateau at the edge of the Blake Escarpment in the dive we had 2 days ago. We collected one.
Yeah, much deeper.
Much deeper at 1,300 meters water depth, and we're currently at 730 meters. And of course, we're under the Gulf Stream now. We were well away from the Gulf Stream before. We were actually, that area was more influenced by the western boundary current. Undercurrent, sorry.
Yeah, Pat, it looks like you're approaching another little dip.
Hey, Doc, this is Zach
like
here.
there's more slope after
I
it.
think I've seen a couple keratoisis bamboo corals, 2 colonies now. If we see another one, could we get a zoom?
Okay.
Sure, Zach, thanks.
Be on the lookout for bamboo.
That might be one in the center there.
Roger.
We have more Synaphobranchid cutthroat eels swimming around.
Several.
So
I see
I'm going to
at
go
least 5 right now in the view.
to the specimen in a little bit farther up. I'll get a better angle on it. Watch these. This is the bamboo that was requested. Straight ahead.
Zach, is that what you're looking for? Right here
Roger.
with the lasers?
Yes, it is.
Okay.
And I'd love to get some other people's opinions on what it is as well.
Okay.
We also have a very interesting sponge that we just went over
in
to the
the
lower
foreground.
left. And most of the coral
Stand
matrix
by
that we've
video.
seen so far has been Analopsamia profunda. We also find Lophelia pertusa in this area, but so far most of what we've seen has been Analopsamia.
Okay, co-pilot, I might need your help with camera here.
You can see several little orange cup corals that are
So let's
growing
go
on
for
the
the one
dead
up above.
Analopsamia coral matrix here.
Go ahead, video. Let's tilt up a little, see if we can get it with the darker background. Make it better for, there we go. Thank you, co-pilot.
Thank you for the zoom. It's not even a bamboo coral, so I'm glad we did that.
Yeah, it didn't look like a bamboo. Any other suggestions of a genus? Beautiful video. Yeah, and Sandra Book was pointing out that so far the corals that we've seen, the scleractinian corals that are forming this matrix, look like it's a mix between Lophelia and Analopsamia, which is kind of uncommon at these depths.
Video, if you're clear. Yep.
what we're looking at is an octocoral, which is different
And
from the
co-pilot,
scleractinian. And
once
it's
we get the specimen
scleractinian
view.
that live in the shallows, or
If we can tilt down.
No, I think
The
that
coral
they
reef.
were referring to the mix of
Was that good for the specimen?
scleractinians.
I know, but the
But,
shallow reef building corals, are they scleractinians?
Yes.
Okay, and do they typically have 6 tentacles instead of the 8?
Yeah, so that
I haven't counted one lately, but
or a multiple of 6.
Okay.
They're hexacorals.
Yeah, let's tilt down,
yes.
and that sponge is right between
yes.
my toes that they were talking about earlier. You might want to bring fog lights on for that.
Yeah, and that's,
That's a great
sponge.
sponge, yes.
Yeah, the lower left. If we could get a look at that, please,
Yep,
pilots.
that's what we're working on.
Great, thanks. And I don't think Shirley Pomponi is with us today, but I think that she would really appreciate getting a look at
I
this.
don't believe we've seen that one.
No, it looks different.
It looks like one we collected during the Charleston Bump expeditions. So, again, under the Gulf Stream north of here. I certainly wouldn't be able to say if it was the same species or even tell you what it was, but looks really similar. You can see the smaller holes for the intake of the fluids, and the larger
Pilot's
two at
clear
the
when
top,
video's clear.
or the outflow.
Let's come on. Carry on.
And they are filter feeding
Fog
particles
lights,
out of the
please.
water, so they're constantly circulating water through their
All right.
bodies.
I'm going to take control back.
And Chris Kelly thought that this is a species of Geodia. And we saw probably very different species within that genus at
Coming
deeper
back to port.
sites earlier in this expedition.
I was just following this little ridge for a little bit. Now we'll
Scott
push on
Harris
ahead.
pointed out that he would love to see a core through this mound to see the history of the mound and the growth of the coral. And I believe that Eric Cortez has actually done a core
Pushing
through
hard
a
over this flat
Lophelia
spot.
mound in the Gulf of Mexico. I don't know
Also,
if he'd
Copilot, remember
care
you
to
turned
comment on that.
starboard.
Yeah, we just corrected that. Do I need to keep coming more? Oh, yeah, okay. Okay, push it forward. Okay. Moving ahead.
Maybe we should introduce at least some of the members of our chat team until we
I think we're
see
ready for
something
another
that
move.
makes us stop
Copy
talking.
that. Let's see, you're
The
looking.
biota
I
seem
might
to
cheat,
interrupt
maybe like
us quite
5-0 this
often.
time. If everyone's okay with that, and I'll
Yep,
keep
5-0.
5-0.
5-0 sounds good.
Go ahead,
So
Nav.
while we're scanning the horizon here, we have Adrian Copeland from NOAA, OAR and OER. We have Amanda Demopoulos from U.S. Geological Survey.
30 meters, bearing 050, speed decimal 2.
Christian Jones, who I'm not sure your affiliation. I don't have you on my list. I'm sorry. We have Chris Kelly from the University of Hawaii, who has joined us on many of these dives so far. Thanks, Chris. Eric Cortez, who is from Temple
ROV nav, bridge
University.
move initiated.
Let's see, Heather Judkins, you're a newcomer, I believe, to our group, so you might want to let us know where your affiliation is. And welcome to the chat room. We also currently have Iris Sampaio from University of the Azores. Azores and Senckenberg am Mir in Germany. Welcome, Iris. Nice to have some fellows from
See if I can't
international
stretch out
locations.
into
We
this
have
area.
a few others. We also have James Murphy, who is the Knaus Fellow
Let's
at
just
NOAA
snap here. Is
OER.
that the skeleton of the coral that's so dark?
That might be actually, it looks like a Liopathes black coral. Good eye, pilot. So they really
like two
black
different
stuff
types there.
is now
Oh, let's
dead
come
black
wide.
coral,
And I'm
and
gonna get
actually
you
it looks
much
like there
closer.
might be Chrysogorgia in the middle of that clump of life there too. That's a very interesting little spot.
Yeah, we're going back into it, watch lead on just repositioning.
Great, thank you, pilot. Then in the back right we have a fish that we can check out after we look at this. Might be a Limonema, but I'm not sure. Limonema. Yep, and this looks like a Leiopathes here, and so you can see the black skeleton exposed where there's no more tissue. And then it looks like it might be another one of those Duva florida that's growing on the dead skeleton to the upper left. And you can see the hydroid and then a little copepod, maybe, or no, that might be a tiny anemone.
Background, that's a little more transition.
That's growing on the hydroid. Okay.
Yeah, there's another one.
There's
amphipod,
amphipod, yeah.
Centering
I think
back
the
up.
other one was a small amphipod.
Okay.
There you go. I don't know. Watch leads, is there any particular areas you'd like to see?
Yeah, if we could just zoom a little bit to the left there, it looks like there's another coral growing on this that's in the left-hand corner. Thank you.
Coming up and left.
And saying hello to Heather Judkins. She's just pointed out that she's from the University of South Florida, St. Petersburg, and she's studying cephalopods with Mike Vecchione. And, oh yes, she usually participates in the midwater dives. I knew your name looked familiar, but I couldn't remember where you're from. Thanks for joining us, Heather. Yeah, that looks like that soft coral that we saw previously. So we have part of this Leiopathes that's alive, and that's the orange tissue that you see in the back. And then this part of the skeleton is no longer alive, and it's been inhabited by a whole bunch of different species, including the soft coral Duvia florida. And hydroid up to the top left. And more hydroids to the right.
So no hard surface that is up in the current gets left blank or empty for very long.
If there's real estate, it will be bought.
That's right. Seller's market.
All right, pilot's clear. Let's go see if that fish is still there.
And while we're looking for the fish, I'll continue with my introductions. We also have Jason Chater and Jill Bork from the U.S. Geological Survey. Let's see.
Oh, yep, right front and center.
And Megan McCullough
from
North Carolina Museum of
Co-pilot,
Natural Sciences. Megan's been with
venture
us
off a
quite
little bit.
often.
Go ahead, video.
Michael Vecchione, excuse me.
Yep, and that's a Limonema.
From the NOAA National Marine Fisheries.
I think that's Barbatulum, if I'm not mistaken.
The National Systemics Lab. We have Nolan Barrett, who is at the Natural Products Discovery Lab in the Drug Discovery Department at the Medical University of South Carolina. Nolan has been on board, and he's the youngster in the group, but doing incredible work. We'll get him to weigh in at some point.
Ooh, and we have a beautiful glass vase sponge there in the lower left, similar to those that we were seeing a couple days ago on a dive. But those that we were seeing were on mud substrate. And I think Scott was pointing out that there are different types of bases of those glass sponges, and some of them have to adhere to something hard, and some of them will send out other stabilizing, I forget what the name of the structures were that he said.
Too much time. Let's come out a little bit so I can get lasers on them. There we go. All right. Time to move on.
Yep, and Sandra Brooke
let's go on this one real quick. I think that's a new fish.
Limonema codling.
Let's snap here, video.
And here's another fish. This is a rat tail, Nizumia species. Nizumia bairdi is a common one. I'm not sure if this is that species or not.
Okay. Unfortunately, I think I need to move on. Let's come on.
And we have Morinellopsamia profunda, the
Got enough
white
for
coral
still
that
there,
we're flying
Naidee.
over now. The hard stony coral.
Continuing on.
And wow, we've got a lot more standing coral skeleton here and lots of sponges.
Roger. There's a urchin, bottom left.
Oh, right. Yeah, we haven't seen that urchin before that you see
How am
on
I
the
doing,
bottom
co-pilot?
left.
Okay. Video. Let's go on this urchin when you get a second. Let's just snap for ID.
And Nolan
ROV
Barrett
nav,
commented
bridge, move
that
complete.
it's a rosellid vase sponge that we were looking at. And Chris Kelly agreed,
All right,
and he
let's
also
come
said
wide.
that the Lamanima species in the Pacific aren't as handsome as our Atlantic species. So
Look at this mound, and then I'm going to go off left for you, co-pilot.
yeah, and Chuck Messing commented that this little orange cup corals that we're seeing might be Bathopsamia, and I totally agree. This is watch one.
Go ahead.
Could we look at that yellow octocoral that's
Yep.
there on the left? You're probably ahead of me and going that way anyway, but
I was.
and Chuck Messing pointed
Go
out
ahead.
that the Lamanima that we just saw is one of the most common species on the Miami Terrace to the south. And I
Go
think we'll
ahead,
be
video.
seeing those quite a bit more as we move up north as well, at least for a bit. And I don't know if this is a Paramaricia species, possibly.
Turns out I wasn't quite down. There we go. How do you like the light there, Bob? You want to try the fog lights? All right, let's go for it.
It looks to me like the coral behind this is actually Lophelia,
That's lights on.
the scleractinian white coral behind this octocoral.
Tilting down. And pilot's clear when video's clear. All right. So I come off. Let's bring the fog lights back off, please. Thanks, Andy. Carrying on.
And as we saw before, as soon as we move off that topographic high that sticks up into the flow of the current, we see fewer corals. We see the rubble of ancient coral, or older dead
Yeah,
coral.
I think we're going to have to make our next... Oh, I'm turning around south. Okay, I need to
We see more of the sediment drape.
really come around.
Yeah, there might be something, if you keep going
There we go.
towards that direction.
There we go. This is what we want.
Yeah.
And here we are back in coral territory. That was fast.
That was, this is definitely the dominant landscape on the highs.
So pilot, you're looking 4-0 right now.
This really gives you a sense of the
Yeah,
piling
let me come
upward
up and
of
look
the dead
Might
coral.
be a
Yeah.
drop after that. We'll see.
Just imagine how many thousands of years it took to make this 30-meter high mound from the flat plains
Do you want
of
to
the
zoom my
seafloor.
sonar out, see if we can see what the trend is?
Yeah.
So watch leads, we're just doing a kind of a orientation check here. I'm coming up off the bottom a little bit to see if we can find in sonar which way is you know, where that tall point, the local tall point is. So that's what's going on right now.
Roger that, pilot.
So I think just following
So
this
the pilot
ridge to
just
my
to
right.
let us know that they lifted off the bottom off
What
the
do you guys
seafloor a
think?
little bit, and they're looking
Yeah,
around
that's my
with
best
sonar
guess.
to try to point out where the highest point is in our vicinity here, and we'll head towards that.
'Cause it,
30.
this is the top of this ridge, so I think it's going to be back towards the north that we'll need to go.
Also in our chat room today is Rachel Bassett, who is at
So you want
the
to go
NOAA
which
NCOS
direction?
lab in Charleston.
I think we'll
And
need
Rachel
to.
has quite a team with her,
Cut
I
that way?
suspect.
There was
They're
a
one
little-
of the expedition centers.
Back to starboard, there
command
was
centers.
a little more slope,
Yeah.
but there might be a drop off after that.
Okay. And I think we should just keep
Sandra
sending
Brooke.
Sirius a little more north.
Sandra has been
Copy that.
very helpful in
just wanted to
helping us
move-
to define some of the dive
see.
locations. Sandra is at Florida State University.
30 degrees,
Scott Harris from the
30 and
College
30,
of Charleston,
or I can just go straight north.
colleague in the geology
Yeah,
department.
either one of those I think is fine.
15 degrees, 30 meters.
We have
15
Steve
sounds great.
Oskevich at Temple University. Tamara Frank from Nova Southeastern University. Let's see, and Tina Molodtsova, who is another one of our international colleagues. She is at the Shirshov Institute of Oceanology
Roger.
in Moscow. Tina's been on every of our dives on the chat room, and we appreciate all your help, Tina.
Thanks, video.
And we have Zach.
RV
Berg,
nav,
who
I
is
understand
my
30
graduate student
meters,
at College of
015
Charleston.
degrees
He
at a
also
speed of decimal
in
2.
the lab with Rachel
Thank you.
Bassett. And working very closely with Peter Etnoyer, who has been on many
So,
of our
I think
chat
this will
room
be good if we.
discussions of the previous
Current's
dives.
definitely coming.
And Rachel Bassett also works with Peter.
Yeah, almost west. So I think if we can get up to the east of this slope, right, kind of hit this ridge, I think we'll find
ROV nav bridge move initiated.
a lot of coral.
Yeah, copy that. Sounds good.
So almost to the point where I'll turn 90 degrees to kind of go along the edge rather than pointing towards the east.
Yeah,
If that
copy.
makes sense. All right. Keep pushing out. There's slope for sure. Tilting up. It's interesting, these channels. I wonder if they get so sedimented that it's harder for stuff to grow. Seems like plenty of current. We'll stop and get a close-up of this urchin while we wait for Sirius.
Pilot, is there a chance we can zoom in on that urchin, or have we gone a little too
That's
far?
what we're going for right now.
Terrific. We got a brief glimpse of one of these earlier that had to move on with the current and with the ship's position. Periodically we have to catch up to the ship. Cheryl just stepped out to go to lunch, and now sitting on my left here is my colleague who is from the Smithsonian. Would you like to introduce yourself?
Yeah, sure.
Go ahead, Vivian.
I was going to. My name is Stephanie Bush, and I work in the Department of Invertebrate Zoology at the Smithsonian.
if you've tuned in just recently, you may not know who I am. I'm Leslie Sauter. I'm at the College of Charleston in the geology department and have the good fortune to be the science geology lead on this expedition.
Looks like, yeah, the distal portion of the